Hello and welcome to Digital Medboard. Here we go. We begin with gray top tube. It is used for glucose estimation. The additives present in these tubes are sodium fluoride and potassium oxalate. Sodium fluoride inhibit enolase in the red blood cells. Since RBCs primarily utilize glucose for energy, if they are allowed to metabolize glucose in the collected sample, it could lead to falsely low glucose readings. To prevent this, fluoride inhibits enolase, which stops glucose metabolism in RBCs and ensuring an accurate glucose estimation. The blue top tube is used for coagulation assays such as prothrombin time, activated partial thromboplastin, fibrinogen assays, and factor assays. The additive used in blue top tube is 3.2% sodium citrate. For instance, imagine it is a black top tube, which specifically used for erythrocyte sedimentation rate (ESR), and it contains 3.8% sodium citrate. The green top tube is a heparinized tube, used mainly for stat biochemical analysis, such as intraoperative parathyroid hormone (PTH) or troponin T (TROP-T) assays. For example, during thyroidectomy, it is crucial to preserve the parathyroid glands. An intraoperative PTH test is performed before closing the suture site. Heparin acts as an anticoagulant, eliminating the need to wait for clot formation. So, the plasma can be quickly separated and used for rapid assays. Heparinized tubes are also performed for genetic analysis, including molecular genetics and cytogenetics, as they yield high-quality DNA and RNA. The yellow top tube contains gel and clot activator. After blood collection, centrifugation causes the gel to form a mechanical barrier between the cellular component at the bottom and the serum at the top. This prevents contamination of the serum by the cellular content, especially important when the sample is stored or transported for long periods. The barrier helps prevent cell lysis, which can otherwise release the intracellular fluid into the serum. The lavender top tube, also known as the EDTA tube, which contains K2 or K3 EDTA. It is used for complete blood count and HbA1c testing. Additionally, it is used for labile parameters like ammonia, aCTH, and PTH, which degrade rapidly due to protease in the sample. EDTA chelates calcium, inhibiting protease activity and preserving these sensitive analytes. The red top tube. A helpful mnemonic is RSS, red, silica, and serum. The red top tube contains silica, a clot activator that promotes clot formation and allows for serum separation. If you found this video helpful, don't forget to like, share, and subscribe to Digital Medboard. Got questions? Drop them in the comments below. See you in the next video. Until then, stay tuned. Choose.